So now that we've seen how graded potentials create action potentials, let's now see how those action potentials cause the release of neurotransmitter at the synaptic cleft. We've already seen this process occur at the neuromuscular junction, and the events that occur here are exactly the same. But let's do a quick review of it. So at the junction between two neurons is a narrow, fluid-filled gap called the synaptic cleft. Now, the action potential that's created in the neuron and traveling along the axon cannot cross the synaptic cleft to another neuron. So chemical neurotransmitters are used to signal adjacent neurons because chemicals are able to cross the fluid-filled space between the neurons. So the cell that's releasing the neurotransmitter is called the presynaptic neuron, since this word presynaptic means before the synapse. The cell that's responding to the neurotransmitter is called the postsynaptic neuron, since the word postsynaptic means after the synapse. So focusing on the presynaptic neuron, we see that there was a stimulus that was created, or that a stimulus that created an action potential in this presynaptic neuron. Now that action potential propagates down the axon of the presynaptic neuron and arrives at the axon terminal, which is the expanded end of the axon, which stores the neurotransmitters. So when it arrives at the axon terminal, the voltage change of the action potential causes these channels, called voltage-gated calcium channels, that are found in the membrane of the axon terminal, it will cause these to open. And so this allows calcium to enter the axon terminal of the presynaptic neuron. So the entry of calcium is important because calcium causes the synaptic vesicles that are storing the neurotransmitters to fuse to the membrane. And when they fuse to the membrane, they release the neurotransmitter that they're storing into the synaptic cleft. Now, after those neurotransmitters are released, they then diffuse across the synaptic cleft, and they bind to receptors on the postsynaptic neuron. The postsynaptic neuron then responds to that neurotransmitter by creating either an EPSP or an IPSP. So recall that EPSPs are excitatory postsynaptic potentials, or you can think of these also as excitatory graded potentials, while the IPSPs are inhibitory postsynaptic potentials, or you can also think of these as inhibitory graded potentials. So when we say the cell responds, we mean that the cell responds by creating an EPSP or an IPSP. So it will respond by creating one of those two types of graded potentials. So now that the postsynaptic neuron has been signaled, any excess neurotransmitter that's remaining in the synaptic cleft is removed by one of two ways. It can be removed by enzyme breakdown. So there are enzymes that are found or that exist on the postsynaptic membrane, and these enzymes break down that neurotransmitter. The neurotransmitter that's in the synaptic cleft, or the excess neurotransmitter that's in the synaptic cleft, can also be removed by a process called reuptake, which is indicated at item 7 here in the diagram. So reuptake is a process in which the presynaptic neuron actually reclaims any unused neurotransmitter, and what it does is it repackages it back into new synaptic vesicles, so that way this neurotransmitter can be released later on. So these are the two processes by which, or the two main processes by which excess neurotransmitters can be removed from the synaptic cleft after signaling has occurred.